Good morning, everybody. Good morning, everybody. I'm sorry. I would like to welcome you to a very exciting R&D webcast around Immunic's multiple sclerosis development program. My name is Jessica Brew, Head of Investor Relations and Communications at Immunic. I will also be the moderator on today's webcast. As always, in the beginning of our webcast, I will walk through some organizational items first. Please note that all participants will be in listen-only mode, and this event is being recorded. During and after today's presentation, there will be an opportunity to ask questions. If you joined the webcast via the Zoom platform, there are two ways to submit questions. You can either submit your questions in writing via the Q&A tool of the Zoom portal, or if you would like to speak with us directly, please use the raise hand function in the Zoom portal to queue your question. Before we begin, I also would like to remind you that this presentation may contain forward-looking statements. Such statements can be identified by words such as may, will, expect, anticipate, estimate, or words with a similar meaning. And such statements involve a number of risks and uncertainties that could cause Immunic's actual results to differ materially from those discussed here. Please note that these forward-looking statements reflect Immunic's opinions only as of the date of this presentation, and it undertakes no obligation to revise or publicly release the result of any revision to these forward-looking statements in light of new information or future events. Please refer to Immunic's SEC filings for a more detailed description of the risk factors that may affect Immunic's results and these forward-looking statements. All right, during the next 2.5 hours, we will provide an update on the scientific, preclinical, and clinical progress of our lead asset, Vidofludimus calcium, and multiple sclerosis. In addition to its mode of action, an update on our ongoing phase 3 ENSURE program in relapsing MS and phase 2 CALLIPER trial in progressive MS, as well as the drug's potential strategic and commercial positioning in the MS landscape. We will also present very interesting new data from our phase 2 emphasis trial in relapsing remitting MS. Moreover, 3 renowned MS experts will discuss recent scientific findings and their effect on the MS treatment landscape. Let me also introduce today's speakers. We warmly welcome our 3 external speakers, Dr. Fred Lublin from the Icahn School of Medicine at Mount Sinai Hospital in New York, Dr. Larry Steinman from the Stanford University School of Medicine, and Dr. Heinz Wiendl from the University of Münster in Germany. We extend our thanks to Dr. Lublin, Dr. Steinman, and Dr. Wiendl for joining us today. Speaking from Immunic are Dr. Daniel Fitt, our Chief Executive Officer and President, Dr. Hella Kohlhoff, our Chief Scientific Officer, and Dr. Andreas Müller, our Chief Medical Officer. All right, I'm more than happy to hand over now to Daniel to kick off the presentation. Please go ahead.
Yeah, thank you, Jessica. And I have to be honest, I'm super excited myself that we got such an outstanding panel together to give us a first-hand account of the latest and greatest in MS research today. But of course, we also want to use this data to show how well our lead molecule, Vidofludimus calcium, fits into this context. Previously, we have already shown excellent data on this wonderful molecule in large phase 2b trial and are now testing it in 2 pivotal studies in RRMS, the ENSURE-1 and ENSURE-2 trials, and also in a phase 2b efficacy study in PMS called CALLIPER. With IMU-935, we have a second oral drug candidate in development targeting chronic inflammatory disorders. IMU-935 is currently being tested clinically in patients with psoriasis and CRPC. We have recently read out data from a group-level interim evaluation of our phase 1 trial of IMU-935 in psoriasis patients. The first 2 doses tested 150 milligram once and twice daily, did not separate from placebo. However, this trial was affected by an unexpected high placebo rate. Our IMU-935 team is working on further evaluation and planning of next steps for the further development of this promising molecule, and we plan to provide an update and guidance on potential next steps in the first quarter of 2023. Our third clinical development program is investigated in potential for its potential of IMU-856 to restore the intestinal barrier function. IMU-856 uses a novel mechanism of action and has already provided very good safety and tolerability data in healthy human subjects, which we published this summer. We are currently evaluating the effect of IMU-856 in patients with celiac disease who are controlling their disease on a gluten-free diet. We expect data from this part of the phase 1 trial next year. But let's switch back to our lead molecule, Vidofludimus calcium. The understanding of multiple sclerosis is still limited, but we know that incorrect activation of the immune system plays an important role. Also, it is now known that Epstein-Barr virus play a role in the development of MS, but probably also in the progression of MS. And then there's another important observation. MS seems to progress continuously, smoldering, even somehow independently of relapses in RMS patients. Vidofludimus calcium has the potential to have a positive effect on these different aspects. The molecule inhibits a protein called dehydroorotate dehydrogenase, DHODH. It is a key enzyme in the biosynthesis of pyrimidines, an important building block in cell metabolism. However, normal cells are using a salvage pathway to generate enough building blocks for RNA synthesis. The target DHODH is therefore only important in such cells where cell metabolism is overactivated. Therefore, inhibition of DHODH does not lead to non-specific immunosuppression, but to a targeted effect against the bad guys in the immune cell population. Mechanistically, our team was able to show that Vidofludimus calcium prevents the secretion of inflammatory cytokines, which play an important role in pathogenesis of MS. A second effect of DHODH inhibition is the associated antiviral effect. We call this a broad-spectrum antiviral efficacy. Viruses such as EBV or John Cunningham virus can convert an infected cell into a machine that produces further virus particles. This process in turn requires a strong supply with RNA building blocks, which leads to hyperactivated metabolism. Inhibition of DHODH in such infected cells specifically leads to an observed antiviral effect. What is not yet seen on this slide is another interesting effect of Vidofludimus calcium that we have found in connection with its potential neuroprotective properties. We want to report on that in more detail at a later time point, together with supportive preclinical evidence.
So Daniel, I'll take over from here. This is Andreas, and I would like to reintroduce the phase 2 emphasis trial to you that we did that we're doing in relapsing remitting MS patients. So since we have new data on this trial that we want to present today, some very exciting data, I think it's a little re-education on this trial, maybe in order. So next slide, please. So this was a phase 2 trial that we did in relapsing remitting patients, the usual exclusion criteria, and a parallel group design between placebo-controlled and several doses of IMU-838. So if you go to the next slide, please, I think it shows you a general outline of the design of this trial. So in the main treatment period, the blinded treatment period, we were testing doses of 30 and 45 milligrams, and later also in a second cohort of 10 milligrams against placebo. Since the key endpoints in these trials were MRI-based endpoints, so we did a MRI at baseline and then every 6 weeks. The primary endpoint and the key secondary endpoints, like I said, were MRI-based endpoints. We were basically looking at preventing new MRI lesions during the main treatment, during the blinded treatment period, for lesions that were not yet present at baseline, and basically looking for an effect that treatment with Vidofludimus calcium can prevent the occurrence of a portion of these MRI lesions as compared to placebo. So this was already reported in 2019, the outcome of these results, and I will quickly summarize them. But I think I also wanted to point out that at the end of this 24-week blinded treatment period, patients had the option to go into a extended treatment period of up to 9.5 years, which was basically an open label period that we thought to employ to get long-term safety data from from these patients as well, but also to follow them for some of the outcomes of the MS disease. And this is today is the first time that we present to you a interim analysis of this open label period that was ongoing for the last 3 years. And I think these are exciting data. So next slide, please. This is just rehashing the outcome of the blinded treatment period and the primary and key secondary endpoints at that point that we already reported in 2019. Altogether, we had treated 268 patients in this blinded treatment period during 2 cohorts. We saw a very strong reduction in the MRI lesions here. The graphic is for the cumulative number of combined unique active lesions. So basically, these are all new MRI lesions that were not present at baseline for these doses. And we found a very strong effect for the 30 milligram dose that we also continuing now in the phase 3 trial. And it was highly statistically significant. Next slide, please. When you put the reduction in MRI activity or suppression of MRI activity into perspective of other phase 2 trials for currently marketed products in MS that basically use the same study design and a very similar or identical follow-up period, you see that the reduction or suppression of MRI activity that we had seen both in terms of combined unique active lesions and gadolinium enhancing lesions it compares very favorable to currently marketed drugs. And we believe this shows that this drug in terms of efficacy, I think is at least on par with the currently marketed drugs. Next slide, please. What we also had seen in this trial and reported in this trial already, is our effect on a marker that's called neurofilament light chain. Neurofilament light chain is a marker for neurodegeneration. So this increases in blood serum and also in CSF, but we measure here in blood serum when you have neurodegenerations going on. So this can be measured in all kinds of different neurodegenerative diseases, like in MS. So a reduction in neurofilament light chain really signals that a treatment can reduce the neurodegeneration or provides a neuroprotective effect in this disease. So we found in our phase 2 trial, the emphasis trial, we found a dose-dependent decrease of neurofilament and a relatively strong decrease of neurofilament for increasing doses. And if you put this into perspective, next slide, please. When you compare this, for example, with other treatments, and we have here for comparison the ocrelizumab data at 24 weeks, so which is the same time point that we were looking at the neurofilament at 24 weeks. After 24 weeks of treatment, you see that the decrease that we had seen from our trial is very much in line with, for example, high efficacy drugs like ocrelizumab. So we also believe that this indicates a very good neuroprotective potential for Vidofludimus calcium that we will be further evaluating in our ongoing trials. Next slide, please. So after this short historical excursion into what the trial was and what data we already presented, I wanted to talk about new data that we have from this interim analysis, from the first interim analysis of the open label extension phase of this phase 2 trial. Next slide, please. So I already said that in this trial, 268 patients had started treatment during the blinded phase. We had a discontinuation during the blinded phase of 14 patients. So 254 patients finished the blinded treatment phase and had the option of going into the open label phase. All of these 254 patients actually participated in the open label extension period that we are discussing today here. in the last 3 years, because basically the open label period is going on for the last 3 years, since 2019, 45 patients have discontinued, of which 10 are discontinued due to MS-related clinical events. And it also includes 3 patients who had discontinued related to the conflict in Ukraine. So at the moment, there's still 209 patients continuing in this open label extension treatment as of the database cut that we did on October 16th now for doing this interim analysis. So we are quite happy that we have a very stable treatment population, this open label phase of the trial. So this interim analysis contains now data for roughly 525 treatment years with Vidofludimus calcium in the open label phase. This does not include the blinded treatment phase. Like I already pointed out, there's a very low rate of treatment discontinuations during this open label treatment. We have now long-term data in this in this interim analysis for 193 patients who had at least the treatment period in this open label treatment of at least 96 weeks, about 2 years. And that's mainly what we're referring to here, also in the efficacy data. But the longest period that somebody is in open label now is roughly 4 years. So we're starting to have these really long-term data in this open label trial. Next slide, please. So we are reporting today mainly on data on confirmed disability worsening. And why is this so important? And we believe is such an important outcome for any drug now, and especially for Vidofludimus calcium. And this basically summarizes a paper, a recent paper that talked about that in the natural history of relapsing multiple sclerosis, we very much focused in the past on the prevention of relapses, because in a relapsing MS disease, these are kind of the leading clinical symptoms. And of course, with a lot of the therapies that are available now, we had really drastically reduced the relapse rate of our patients. And it was really expected that if you turn off basically these relapses in this relapsing phase, that you get more or less no disability worsening. Because I think the traditional view was that in relapsing disease, the disability accumulation is connected to relapses. But what we had observed now over the last years is that even if you have no relapses, there is a slowly increasing disability accumulation, even in these patients that have basically no relapses. And it was termed silent progression now in the MS community. Next slide, please. And there was always the question, what really contributes to this silent progression? What is this silent progression? And there was a landmark paper basically by a group around Fred Lublin. And I'm very happy that Fred is here today and will report these data that basically looked at what is connected to this disability worsening in relapsing disease, and also, of course, in progressive disease. And it was found, and we'll see more of the data, that this relapse independent accumulation of disability or neurological deficits really plays a large role even in relapsing disease. And here on the right side, I think it just looks at that maybe for the long-term outcome of the patient, it is really more important how a drug or a treatment contributes to slowing the disability accumulation, the silent progression over time, rather than basically preventing relapses. Relapses are relatively rare events nowadays. So the contribution to this silent progression, that relapse independent disability accumulation for the outcome of the patient, really is very important now. And that's why we focus on basically data that we have now with regarding disability accumulation. So next slide, please. So what we did so far from the phase 2 trial, we looked at the blinded treatment phase, the main treatment phase, the first 24 weeks in this trial. And all we were able to report at this point were basically mean changes of EDSS, which is the neurological score that measures disability or accumulation of disability. And we had shown that under treatment with any of our doses of Vidofludimus calcium, we saw a stable mean EDSS for the groups, whereas in placebo it increased. And then we also looked at unconfirmed EDSS progression, basically events where you have an increase or time points when you have an increase in EDSS score, basically this disability score. And you also see that placebo had a higher rate of these than during treatment with Vidofludimus calcium. However, I think these data had to be always looked at with a little caution, because relapse activity and single point events may influence this evaluation. So I think we were aware that you really had to go to confirmed disability worsening, which is usually measured in larger and longer trials, like in phase 3 trials, as the primary outcome for disability worsening. And with this analysis, with this interim analysis now, we're able to present to you the confirmed disability worsening events for the first 24 weeks of blinded treatment, because we needed that follow-up. And I will explain this in the next slide. How do we measure confirmed disability worsenings? Sorry, but I think I should, before you see the data, explain a little bit how this is measured. So you have, let's say, a baseline value of EDSS, and then you have a trigger event somewhere in the trial. A trigger event is basically a protocol-defined increase in the EDSS score as compared to baseline. This is the trigger event, and these definitions for these trigger events are now universal for all of the clinical trials. We're all using the same definitions. But then you have to be aware that this could be a single point event, or it could be relapse related, or it could be related to an infection in this patient, to fever in this patient. So there are a lot of reasons why a EDSS can be increased. So we usually require, and regulators require this as well, that we have a 3 months or 6 months confirmation event. That means that the same increase in EDSS is still present 3 months, in our case 12 weeks, or 6 months, in our case 24 weeks later, so that it is a true disability worsening for this patient that is maintained over time and has clinical importance. And that's what we call confirmed disability, or CDW events. And that's what I'm going to present to you in the next slides from our phase 2 trial. So these are now the confirmed disability worsening events in our blinded treatment period, the first 24 weeks. Since we have all of the follow-up data now in the open label period, we were able to see which of the EDSS increases that we had during the first 24 weeks really had been we were able to confirm 3 or 6 months later. And I'm very happy to present that we've seen a 1.6% of 1.6% of the patient had a confirmed disability event in the Vidofludimus calcium treatment groups, and 3.7% in the placebo groups, which indicates a initial signal that Vidofludimus calcium may either delay or prevent these confirmed disability worsening events. So this is something that is a very encouraging signal. This is something that is a key secondary endpoint in our phase 3 trials, and you need a much larger population to have a statistically meaningful analysis. But I think the data here that we have from our blinded treatment period really are going in the right direction, and confirm this our hypothesis that this drug is particularly good in preventing disability worsening events. So these patients, of course, after the 24-week period, went into the open label period. And if you go to the next slide, these are the data from the open label period now. And it basically looks at the proportion of patients that are free from from 24-week confirmed disability worsening. So that this number is still very high, I think, is a very good thing, because it means most of the patients that have done 1 or 2 years of treatment, so 96 weeks is about 2 years of treatment, did not have any confirmed disability event yet. We only have one line because, of course, there's no more placebo controlled in the open label period. So this basically, we're following these patients, how quickly they accumulate these confirmed disability events. So this is over a 2-year period for 24-week confirmation. So to put this into perspective, what this means really, we don't have a control arm, but we have historical controls. So if you take our number, in this case here, for the 12-week confirmed disability worsenings after 2 years, and compare this with historical trials that also have measured 12-week confirmed disability worsening after 2 years. And these are marketed drugs that have reported this either in trials of relapsing remitting MS or in secondary progressive MS, how many confirmed disability events you had after 2 years. It really shows that Vidofludimus calcium data are in the lower range of historical data. So I think this also is very encouraging from our end, because I think, like I said, I think we always said that with the properties that this drug has, anti-inflammatory, neuroprotective, antiviral, the main advantage of this drug should be in preventing confirmed disability worsening. And at least our data show that the data that we find in the open label phase are in the lower range of historical data. So next slide is basically the same data just for the 24-week confirmed disability worsenings. Also after 2 years, a little bit different numbers, but I think the same meaning again, that I think the Vidofludimus calcium data are really in the lower range of historical trials. And I think these data from the open label really have to be considered a very encouraging outcome, that only very few patients really accumulate these confirmed disability worsenings over time in our open label phase. Next slide, please. So I think we believe that our data that I've just have shown you are really resonating very well with certain new knowledge that we have has been published this year regarding MS as a disease and what's behind the pathophysiology of MS. And we really have identified 3 papers that are the main papers to my mind for 2022. And we are extremely happy that the main authors for all of these 3 papers are here today to present the data. So one of the really, I think, main and landmark papers of 2022 were an analysis by a group around Fred Lublin with an analysis of more than 35,000 patients in MS trials. And they reanalyzed, basically retrospectively, the relapse, the disability events, and really saw whether they contribute more to relapse or whether they're relapse independent. And I think there was quite a surprise that the disability accumulation over time, even in relapsing disease, has a large component of non-relapse, being non-relapse dependent or non-relapse related. And I think this really addresses the question here, this paper and this analysis: what is the contribution of non-relapse related disability to the MS disease progression, even in the relapsing disease? And I'm very happy, if you go to the next slide, that we have Fred Lublin here with us today. He, of course, is very well known in the MS community because he has been involved in the development of new treatments since the early days of treatments, have been involved in all of the treatment advances that has been done in MS. He is very well recognized, and we're very much looking forward, Fred, to your presentation. And I have to say, thank you again for being available here and give the presentation about your analysis. And if you can please unmute yourself, we're ready to go with your presentation.
Okay, can you hear me all right?
Yes, we can hear you.
Okay. I guess you're going to control the slides. Can we make them larger? Or do you, because I don't have a control here.
Yeah, we were working on this, so I try.
Okay, while they're working, so I'm going to discuss some current concepts on how we're viewing the clinical courses of multiple sclerosis, and particularly how disability and impairment develops, because that's become a major unmet need in how we view the disease. And if we get to my next slides, I'll show you disclosures. Which, boy, I can barely see this. Ah, that's better. Okay, so those are my disclosures, shows you how many companies are interested in MS research. Next slide. And so first you need to understand the mechanisms by which MS produces impairment. Clinically, there's two ways. One is incomplete recovery from exacerbations. Because remember, the majority of individuals with MS have relapsing remitting MS. It means they have an acute attack. That attack occurs over days and such, and then improves over the next weeks to months, anywhere from 0 to 100%. If you don't improve, and about 50% of people don't improve, then you've got this incomplete recovery, which causes a stepwise worsening. This is particularly prominent early in the course of MS. The second mechanism for worsening is the gradual progressive worsening that's independent of the relapses that occurs when people enter a progressive phase of MS. Pathologically, we like to think that the incomplete recovery of exacerbations is due to inflammatory activity, and the more gradual worsening due to neurodegeneration. But we have pretty good data that inflammation is involved all along the course of MS. So that particular dichotomization is probably an oversimplification. Next slide, please. And so one of the things we're wrestling with now is of course, defining terms, because how you define a relapse determines how you define the percentage of individuals who are progressing independent of relapse activity, or this PIRA. And so there's still questions on how independent that is from the relapse and which activity you should utilize. So there's been a few papers on this. The paper I'm going to go into some detail that we published in Brain in September, we used a very stringent definition for PIRA by using a less stringent definition of what was a relapse. And then this concept of silent progression that Andreas mentioned, I was just, and I don't know if Heinz Vidofludimus was there as well, but I was just at a session of a meeting I'm at right now in Italy, where they said, if you think your patient has silent progression, you're not listening. And I think that's right. It's not silent progression, it's unrecognized worsening. And it's something that it's good to bring to the attention of clinicians because the patient is going to be aware of it. Next slide, please. And then when we actually look at progressive disease, so what you see here is the square waves are relapses. And this is occurring over time, and the time could be anywhere from separating them from weeks to months to years to decades. But the line going up is what you see in progressive disease, which is impairment over time, worsening. What's not clear if you put a microscope to that, whether it's little micro exacerbations or whether it's truly gradual worsening. And the analogy is in physics to whether it's behaving like a wave or a particle. Next line, please. And so we have relapsing forms of MS, which include the first attack, which is called the clinically isolated syndrome. And as you can see in that first graph, As you get farther along, the relapses don't go back to the original baseline, but are showing stepwise worsening of impairment. That's still relapsing remitting MS, even though they're getting worse on exam. In progressive MS, like secondary progressive MS, the worsening occurs independent of the relapses, but there can be intercurrent relapses in addition to the gradual worsening. Primary progressive MS is what we call progressive from onset, which is it doesn't start with an acute attack, but rather this gradual worsening over time. But even then, there can be superimposed relapses. The biological differences between relapsing disease and progressive disease, again, we think relapsing disease is more inflammatory, but we're not entirely sure of all the mechanisms that are around pure progressive disease. Next slide, please. And so in addition to having relapsing remitting and primary progressive and secondary progressive and the clinically isolated syndrome, which is the first attack, we further characterize the agents as the disease types as to whether they're active, meaning have they had a new attack or a new MRI lesion over some period of time, which we say should be at least measured annually. And also in those in the progressive form of the disease, whether primary progressive or secondary progressive, we want to know whether they're progressing, that is worsening, again, towards it framed in time, but at least once a year. Next slide. And so over time, you see this accumulation of worsening, both through the stepwise mechanism and the gradual worsening. Lesion load goes up over time. Brain volume goes down due to atrophy. We think the amount of inflammation decreases over time, but never really goes away. And then there's a neurodegenerative component, which may at least initially involve the innate immune system. Next slide. So is all MS progressive? And I just got done with a debate about this. So not as we currently define progression. We have some ideas that MS starts earlier than we ever thought. Sometimes before there's any recognized clinical event, you could pick up MRI changes, which occur when someone gets an MRI because for some other reason they got hit on the head, they had a bad headache, and the MRI looks like MS. And then the other question we want to answer was, does it really matter how one reaches a disability outcome, whether it was through relapses or through progression? And we were able to answer that question. Next slide. And so this is the paper in Brain that we were talking about. Professor Wiendl, who's on this call, was also an author on this paper. And what this was Novartis Pharmaceuticals had an enormous database of clinical trials, like 200 clinical trials, that they turned over to the Oxford University Big Data Group to do some deep dives and AI on the data. Next slide. And what they had was, and this is the paper that just came out in Brain in September. Next slide. What we have here is 27,000 patients followed for up to 15 years, 23 clinical trials, 15 were randomized clinical trials, and some were observational, and there's over 200,000 EDSS transitions. EDSS is the major tool we use for measuring disability. There's other tools that one can use, including adding on to things to the EDSS, but that's what we did for the assessments I'm going to show you. Next slide. And so the things that we were looking at, our objectives were, what was the impact of relapses on acquiring disability? What is the role of progression independent of relapse activity, this PIRA? What are the main risk factors for accumulating disability? And what was the impact on treatment on the time to reach milestone disability levels? Next slide. And so these were the two mechanisms we're looking at. The raw or relapse-associated worsening is an acute attack which you look at the square wave on the left, and then it goes down, but it doesn't get back to the original baseline. So all of that damage, that worsening in the metric, in this case, the EDSS, was due to residual from the relapse. On the right-hand side, you have the PIRA, which is the progression that occurred outside the window of a relapse. And what we need to determine better, we did 90 days here, then plus 30. We need to better define what that window is. But we looked at the relative contributions of disability accrual, which is the raw versus PIRA, and looked at when PIRA starts in the disease course. Next slide, please. And so first we said relapses, and this has been an argument going on for a long time, is how important are relapses? Well, we know that our current therapies are very effective at stopping relapses. And the question is, does that make a difference in terms of their ultimate impairment or disability? And what this chart shows, if you look at the 6-week confirmed disability worsening that was discussed by Andreas, if you had a relapse, the AR1 in the year prior to entry into the study, or AR2 2 years prior to entry into the study, then you increase the risk of a relapse, at least in 1 year, by almost 50% and about 40% in the full database. When you look at the phase 3 studies and even the placebo-controlled studies, you see something fairly similar. So relapses increase the risk of future disability, and it increases that risk by about 31 to 48%. Next slide, please. And so what is it? What's different? Well, clinically, they're different because the relapse is an acute event. The patient's well aware of it. They complain of something new, whether it be loss of vision in one eye, numbness, tingling, weakness, bladder difficulties, and such. That, again, occurs over hours to days, and it can last from days to weeks to months to decades without improving, but most of the improvement occurs within a couple of weeks, but there's still a tail to that it can go out much longer. And again, pathologically, what we're seeing is demyelination, inflammation, neuronal and axon loss. Immunologically, we think the adaptive immune system is involved more with relapsing disease, and the innate immune system with progressive disease. But my other two colleagues who are on the call can tell you more about that. They're more facile with it than I am. Next slide. And then we look to see whether it mattered how you got to various disability levels. So here we're looking at EDSS score of 1 means you find something on the exam, but the patient really isn't aware of any impairment. EDSS of 4, you're starting to have some trouble walking. And EDSS of 6, you need a cane to walk. So those are the different hard outcomes that we looked at here. And what we found were two things on this slide. One was, if you were being treated, then you were less likely, it took longer to reach those goals. So if you look, for example, to go from 1 to 4 in placebo-treated patients was almost 9 years. And if you were treated with a disease-modifying therapy, and there was a number of different ones that we looked at, it was almost 12 and a half years. And to go from 1 to 6, again, you're gaining several years. Going from 4 to 6, you gain a little less, but you still gain. On the right-hand side, we looked at how you got there. And so if you go to the far right, the relapse-associated worsening took 3.87, almost 4 years to go from 1 to 4. And the PIRA without relapses was a little better at 3.5, a little over 3.5 years. But if you had PIRA and you had separate from the PIRA relapses as well, then you hit those milestones much faster. So turning off the inflammation of relapses and slowing the accumulation of disability delays the accrual of difficulties. And that can be seen across the board there. Again, least common when you're going from 4 to 6, because now you're already getting individuals with significant residual impairment. Next slide. Now, here was where this is where some of the surprise came in. And I think my friend and colleague Ludwig Kapos was the first to describe this, which is if you look at the boxes on the left, relapsing remitting MS, the red is in a clinical, this is in the whole data set at the top, and on the bottom is the phase 3 trials. So let me start with the phase 3 trials, because that's the one where we have the most comprehensive assessments done. So in the phase 3 trials, you can see in the red, almost 36% of individuals in the study had a relapse, and the blue represents those who didn't have relapses. Now down below, you see the relapse-associated worsening as measured by 6-month sustained disability worsening. Sustained meant that once they hit the worsening, the confirmed that Andreas discussed, they never get better. And so it's a more stringent measure of progression independent of relapse activity. And that's what you see in the green bar. And so the green bar was the PIRA, the brown bar was the relapse associated worsening, and there's about equal percentages of each. Now it's still a very low percentage in these relapsing studies were early MS, and it's only like 3 to 5% of the total population. But that 3 to 5% of the total population was sort of a surprise to us that was going on. And that's where this idea of unrecognized worsening is coming from. Now, if you look at the observational studies, the full data set, you see an increased percentage in PIRA, and there's others who are reporting even higher numbers than we have there. But part of that is because in the observational data sets, there's not standardized visits, and therefore relapses could be missed. Now, when you shift to the right to secondary progressive MS and primary progressive MS, then the progression outside of relapses becomes much, much more important. And that's one of the difficulties we have in treating those diseases is that's harder to treat. Next slide. And here we look at treatment effect. So the top row is the treated individuals with a variety of different disease-modifying therapies. The bottom is placebo, and we're looking at the same metrics. And what you see here is that, of course, in the placebo patients, there's many more relapses. The red area is much larger than you see above it in the actively treated, because that's what our therapies do. They take away relapses. But one of the other things you see is in the active treated group, there's more PIRA than there is in placebo treated groups, because even though there was a reduction in the percent of treated between active and placebo, the degree of contribution of PIRA to the overall worsening was greater because we don't treat it as well as we do the relapse associated worsening. And then you see the same thing going across for secondary progressive MS, primary progressive MS, where we don't have as good tools to treat. So we have very modest treatments for both those forms of MS. And you can see that from the graphs that there's not much difference between the active treated group and the placebo treated group. Next slide, please. So the key conclusions from the study were the relapses in 1 year prior to entry into a study increased the chance of disability accumulation by 31 to 48%. PIRA starts early. That's about 50%, a little less than 50% of the disability worsening in RRMS. But remember, the amount of disability worsening in early RRMS is a small number, which is one of the reasons why a number of the studies, when they succeed in reducing the annualized relapse rate, don't necessarily succeed in reducing the accumulation of disability because the number's too small. As you get into secondary progressive MS, primary progressive MS, PIRA is the main driver of the disability and impairment. What I didn't show you the data was the principal risk factor for disability accumulation were disability coming into the study and older age. And then using disease-modifying therapies delayed disability accrual by years, with the potential to gain time being highest in the earliest stages of the disease. Next slide. And so when we think about MS in sort of a holistically way, it starts with this RIS, or radiologic isolated syndrome. This is individuals who get an MRI scan because they got a bump on the head or a bad headache, and it looks like MS. And when we study them, two things happen. One, the majority, when they switch, develop the first attack of MS, which is CIS, or relapsing remitting MS. But a small percentage of them, like 10%, go on to primary progressive MS, which is the same degree of the percentage in the general population of primary progressive MS. And so we think that this radiologic isolated syndrome, the early MRI changes, are what ties together the various forms of MS. Next slide, please. And then we think of MS as one disease, whether it's starting as MRI onset or relapse onset or progressive onset. And then we can further characterize it as to whether it's relapsing remitting, primary progressive, relapsing remittings may go on to secondary progressive. And then we look to see whether they're active or not active over time, and progressing or not progressing over time. Next slide. So our next goal was to have more biologically based clinical courses of multiple sclerosis. And we're not there yet, but we've been calling for the courses, the descriptions that I gave you are what you see when the patient's sitting in front of you. But we're hopeful that we'll be able to get more information from advanced MRI metrics. Although right now you can't use the MRI to separate a relapsing remitting patient from a progressive patient. But in the future, looking at solely expanding lesions and paramagnetic rim lesions, maybe we will. And certainly looking at atrophy may be of some value in prognosticating. We also want to look at fluid biomarkers. You heard discussion about neurofilament light. That one's been a bit of a disappointment because we had hoped it would tell us more about progressive disease and inflammatory disease, but it's turned out to be the opposite. But there are other biomarkers like GFAP, and we may need a composite to help us prognosticate there, and ultimately to give us a goal on what the underlying pathology is. Is it primarily inflammatory? And if it is inflammatory, is it adaptive immune or innate immune? And can we target them? Is there demyelination? Is there potential for repair? Is there neurodegeneration? So there's lots for us to learn once we get a better handle on the biological underpinnings of the different disease courses. Next slide. And this is my crew in Central Park in New York City with their families. And I thank you all for your attention.
Thank you, Fred. This was, I always like that you are able to bring down and very nicely explain a very complex study like this, and with lots of different things that you looked at, but I think really focus on the main outcomes. Thank you, Fred. This was wonderful. We will have the question and answer session then together for all of the experts. So we will go on with basically our next speakers on go into the next topic. We probably wanted to go a little bit more into the importance of viruses for the MS disease, and also in terms of looking at Vidofludimus calcium with its properties in the antiviral area, the antiviral data that we have. Next slide, please. And of course, there was, and everybody who is involved in MS has seen this paper on basically a patient, on a military population in the US, where you have long-term follow-up data, health data, which established a link of a late Epstein-Barr virus infection with the onset of MS. So, and we will not go into this here in this further, but I think that is now has been thoroughly discussed in the MS community. Next slide, please. And it is also connected. So this paper was basically, this US military paper was connected to the onset of MS, but we also have some evidence that maybe Epstein-Barr virus has some influence on ongoing disease during the disease, not only during onset. And we have this is just an example of such evidence that we have seen, for example, that patients with more MRI lesions that are visible on MRI have higher Epstein-Barr antibody titers, for example. So there are several of these evidence that maybe are not conclusive by themselves, but at least increase the evidence that ongoing EBV virus reactivation may play a role. And this is in the next slide, please. There were several data now that looked at basically evidence of EBV reactivation in MS patients. So what you can do here is, for example, you can look at EBV virus DNA shedding in saliva, which should be an indicator of EBV reactivation. So we've seen that there are a number of patients, MS patients, show this virus shedding in saliva during the disease. So this is, of course, fluctuating in these MS patients as well at different time points. Some are shatters, some are non-shatters over time. And this basically is evidence that this maybe that the reactivation is contributing to it. And here on the right, so you see another publication where somebody looked at the number of patients or proportion of samples that have a positive EBV DNA in saliva, and comparing this between a drug that has antiviral effects like teriflunomide and in trials of drugs that don't have an antiviral effect. And of course, this is not the same trial. This is a comparison across trials, but I think here there's some evidence that maybe some of the antiviral treatments like teriflunomide can decrease the number of samples that have EBV shedding. So maybe it decreases the time or that patients have EBV reactivation. So next slide. And this was a transition basically to our own data. So I'll hand over to Hella for these data.
Yeah, thank you very much. So we and others have long known that the inhibition of DHODH by small molecules and thus impacting the metabolism in virus-infected cells leads to antiviral activity. Since DHODH is a host-based antiviral, or targeting DHODH is a host-based antiviral approach, DHODH inhibition should be effective against the various pathogenic viruses. Here we have demonstrated this antiviral activity for Vidofludimus calcium in cellular assay systems against various RNA and DNA viruses, such as SARS-CoV-2, hepatitis C virus, cytomegalovirus, HIV, and arenavirus causing Lassa fever, and recently as well against Epstein-Barr virus. Epstein-Barr virus is a gamma herpes virus and has a special feature of remaining in the body for life after initial infection of epithelial cells, remaining mainly in certain B cells in the so-called latent state. Under certain circumstances, which are not yet fully understood, the virus can be reactivated in these B cells and new virus particles are produced, which can then infect epithelial cells again. This can be measured, for example, as virus shedding from saliva. Vidofludimus calcium inhibits the replication of the virus during the initial infection with EBV, but what is much more important here is that it inhibits the reactivation of the virus and thus prevents the production of further virus particles that would trigger a new immune response. This is because with each reactivation and cycle of infection, a newly generated human immune response carries the risk of new cross-reactive antibodies through a process which is called somatic hypermutation. We will learn a lot more later from Dr. Larry Steinman about cross-reactive antibodies and their relevance to MS. We at Immunic think that blocking the recurrent reactivation cycle of EBV by treatment with Vidofludimus calcium could therefore provide long-term benefit by reducing the ongoing neurodestructive trigger of EBV. And then this is a scientific presentation here. I'm pleased to share with you the experimental data from our collaboration with Professor Marshall from Erlangen, showing that Vidofludimus calcium exhibits concentration-dependent inhibition of EBV reactivation in 3 different relevant cell systems. And with this, I would like to hand over back to Andreas.
Yes, thank you. I just wanted to follow it up with that some of the antiviral activity of Vidofludimus calcium also has been shown clinically. Here on the left side, there is a data summary from actually the phase 2 trial that I had discussed earlier, where we looked at the overall number of infections, but also specifically about the COVID-19 infections in these patients that either were in the placebo treatment arm or were in the active treatment arm with Vidofludimus calcium. And I think here we see at least a signal that there's it prevents maybe, or it may prevent COVID-19 infections if in these phase 2 data. And I think that's likely part of the antiviral activity that we see clinically now, if these patients basically take this drug for addressing their MS disease. On the right side, I think it's another thing that we also found in our COVID-19 study that we did with Vidofludimus calcium, which is very important in pointing out the selectivity of this effect, that we have maybe this clinical antiviral effect. However, we do not interfere with the humoral response of the body, the antibody development against, for example, a SARS-CoV-2 infection. When you look at how many patients, the proportion of patients that develop IgA and IgG antibody titers against SARS-CoV-2 after the infection, we see that Vidofludimus calcium has no influence on that humoral response. So I think it's the best of both worlds, that you have an antiviral effect, but you don't interfere with the antibody development that the body has against, for example, viruses. Next slide, please. And this brings us to the next speaker and to our next presentation. There is, this was a very fascinating paper for me to read, I have to admit, because I think it pulls together some of the things that we already knew about MS disease for a long time. For example, the oligoclonal bands, so some antibody bands being in cerebrospinal fluid available, and then the current EBV data. But I think at least it provides a first hint in how this all ties together: the EBV infection, the ongoing shedding of DNA, the antibody bands that we have found in cerebrospinal fluid for a long time. And this is a paper an investigation that was done at the Stanford group and around Larry Steinman. And Larry has been also very well known immunologist in the MS area, with a lot of very valuable contribution to the MS world. I think he has been very instrumental in the development of natalizumab, at that time one of the high efficacy drugs, and has been contributed a lot to the understanding of the immunological understanding of MS. And I think this paper is really addressing, or starting to address, if there's a connection between the ongoing EBV activity in the MS patients and neurodegeneration. So Larry, I'm very happy that you joined us today, and we'll give a little summary of your investigations that you have done. Very interesting, very enlightening for me, and I'm very much looking forward to hear this again, and in your own words. And also, I would like you to unmute yourself and basically present your data. Thanks for participating.
Thanks, Andreas. And I'm delighted to be speaking to the group today about a subject that every time I look at it, I get more excited. I think we have a lot ahead of us to try to understand. So let's proceed to the next slide and dispense with my mugshot. So as Andreas told you, there was a major paper that came out from the Esquerro group at the Harvard School of Public Health. And he used an extensive military database from the US military on 10 million young adults on active duty. 955 were diagnosed with multiple sclerosis, and approximately yearly, including on entry to the military, they would have blood specimens taken, and they were banked, and the bank was accessible to researchers after a considerable amount of screening. So the headline in this paper in Science was that the risk of MS increased 32-fold after infection with EBV, but was not increased after infection with other viruses. They concentrated their focus on the other virus story, mainly on CMV, another virus from the herpes family. In the next slide, we'll get into a little more of the data. So the editors at Science asked me, and I asked my colleague to join me, Bill Robinson. You'll hear more about Bill, who's the chair of the rheumatology division. So as I said, there's over 10 million people who were studied. Of the 900 or so who had MS, 801 specimens actually were available in the bank for serial testing, and 800 of them were EBV positive. But one of the big conundrums is that of all the people in the room where you are, or on the internet where we are across the world, greater than 95% of us have positive serologies from EBV. We get kissed by our mother, we get kissed by our friends, and this virus spreads in saliva. But they had a very interesting group in this study. 35 patients, 35 people with MS entered the military seronegative and without known MS. And of the 35 who went from seronegative to seropositive, there were 34 of the 35 who converted, and they converted before the diagnosis was made, and their levels of neurofilament light chain went up accordingly. So that was further compelling evidence, because when a professional epidemiologist says that this virus with a 32-fold index of stimulation is the trigger for MS, it better be very solid, and the data here are magnificently solid by statistical analysis. Let's go to the next slide, please. So embedded in the data was a very interesting finding. They had some expert virologists in the group at the Harvard School of Public Health, and they did a viral scan looking at what regions of the Epstein-Barr virus, its various proteins, including its transcription factors, EBNA1, 2, and 3, envelope proteins, and capsid proteins. When you looked at serum antibodies, what did they bind to? So at the top of the list was a region between residues 365 and 420 in EBNA1. And what I'll tell you about shortly is our research independently had taken us to that region. But a few other groups across the world, their research also took them to that region. So for instance, Thomas Olson and his colleagues at the Karolinska had studied a chloride channel known as anoctamin 2. Their strategy was a little different than our strategy. I'll tell you about our strategy in a second. But that's within the 365 to 420 region. Another group at Oklahoma Medical Research Foundation also found cross-reactive antibodies to myelin basic protein between residues 411 and 426. So I'd like to call this, for want of a better description, a hotspot of molecular mimicry. This general region of the Epstein-Barr EBNA1 transcription factor doesn't have much secondary structure. And that's interesting in itself. That occurs in a lot of molecules or portions of molecules in nature. But we'll just have to keep this in mind because when you look at that p-value associating it with MS, there's a lot of zeros before you get to a number, 0.00073. And you can see there's another EBNA1 hit and an EBNA3 hit. Of interest because Epstein-Barr vaccines are being developed that include some of the external proteins. There's also hits with the capsid protein and envelope glycoprotein. So we'll have to keep that in mind because I'm now going to tell you about something that we need to think about in the design of vaccines, because maybe there are certain regions in EBV or its transcription factors that it might not be wise to create an immune response to. So let's go to the next slide, please. So this is a cartoon from the perspective that Bill Robinson and I wrote about the Escarrillo paper. It was published January 13th. And we talked about some of the features of the Epstein-Barr virus that may be important in pathogenesis. And we put at the top of the list molecular mimicry. Now, we were a little bit constrained in writing this perspective because of the complexities of peer review. Our paper was not yet out, and we couldn't discuss our paper, even though it came online 2 days later. So just as a sidelight, we had to put it on a file server as a preprint, and then we could talk about it because it was a preprint. Such is modern publishing. That's not germane to the science involved here, but it's an area of modern amusement as far as I'm concerned. So molecular mimicry, I'll talk about that a lot in a second. B-cell transformation, the Epstein-Barr virus infects our B cells, and then it becomes latent. And there's two major latency proteins, LMP1 and LMP2. Then there's the issue of CNS tropism. So Epstein-Barr virus infects CD20 cells. We know that there is probably immune surveillance, and that there's the development of oligoclonal immunoglobulin in the cerebrospinal fluid of MS patients. And there are also some meningeal nests of lymphocytes. So in that way, a CD20 B cell can get into the central nervous system, and it may remain happily there. In the Nature paper, we actually show that one of the main homing molecules that allows the CD20 cell to get into the central nervous system, alpha-4 integrin, is actually lost or diminished by more than a log once the CD20 cells get into the CNS. And then there may be other mechanisms. Let's go on to the next slide. So the paper that came out 2 days later, I'll show you pictures of the authors in a second. The first author is a brilliant young neuroscientist named Tobias Lanz, who came from Michael Platten's lab in Heidelberg. He came to our lab 14 years ago, went back to Heidelberg, finished his training. And the other authors on this paper, it was an immense effort. I count over 30 individuals. Peggy Ho was one of the people who trained him at the bedside. And the senior author is my long-term colleague, Bill Robinson. Now, we mentioned in this paper, can we eradicate MS by targeting EBV? And, you know, that's something that has to be proven by further experiments. So could this be done with an antiviral? And we just heard some very intriguing data about your proprietary compound that has antiviral activities. I mentioned the EBV vaccine. There's a lot of work going on with immune tolerance. My lab is involved in it. And a small company in Mainz, Germany called BioNTech, I say small company with irony, because they published a beautiful paper in Science using the RNA technology to tolerize to what I'm going to be talking about shortly. So, and there may be other approaches that I'm not covering here, but the field is open, and we need to know if getting rid of EBV in the nervous system or getting rid of these cross-reactive molecules, so-called molecular mimics, by immune tolerization will help tame the disease. Next slide. So here are pictures of Tobias and Bill and Peggy. We've all been working together for a very long time. And in the cerebrospinal fluid, there's a collection of plasmablasts. You don't see them concentrated in the blood. And the plasmablasts are IgG. Now, there's another complexity here, because in the spinal fluid, there are three very well-known types of clonal antibody expansions that represent our response to childhood infections, or more recently to childhood vaccinations, to measles, rubella, and varicella zoster. So the MRZ monoclonals are there. In addition, there's a substantial population of clonally expanded, somatic hypermutated antibodies directed to the Epstein-Barr virus. So we pursued not the MRZ reactions, although we have evidence in the Nature paper, but we pursued the specificity of the antibodies in the spinal fluid to Epstein-Barr viral components. So let's go to the next slide. So we had some amazing tools at hand. We had solid phase viral scans of a great portion of the known human virome. We added features to it from various EBV components. And then we had a human proteome chip that has about 16,000 human proteins. And we asked, so the clonal antibodies that we expanded in dishes in our lab, what did they bind? And at the top of the list, there in red, is an example that some of the antibodies bind a molecule called GlialCam, glial cell adhesion molecule. This is a molecule primarily expressed in the central nervous system, and there is some expression in the liver. That's interesting because to our knowledge, the liver is not directly involved in multiple sclerosis, but GlialCam is there as well. And there's another interesting feature of GlialCam. It's a chaperone of a chloride channel. Now, I mentioned earlier that the Karolinska group, Anoctamin 2, is a chloride channel. So there's something about chloride channels that are emerging in this very interesting story. Another interesting aspect of GlialCam, it has remarkable protein homologies if you do blast searches to perinodal proteins such as contactin 1, contactin 2, neurofascin, and so on. So that's interesting. There's a very rare genetic disease, I've never seen it, involving the white matter. But GlialCam now is on the map, along with some of the more traditional myelin molecules that we've been paying attention to for so long. But this is the first time, in my knowledge, it may have been in other people's knowledge, that I ever worked on something called LeoCam. Next slide. So how prevalent are the antibodies? We have not done a detailed study of how prevalent they are in the cerebral spinal fluid, but if you look at anti-GliaCam reactivity in the blood of individuals with MS, roughly speaking, it's about 25% of people at any one time with MS have an antibody to GliaCam. The Anoctamin story, it's about 14%. So even if they're additive and not overlapping, we're still only talking about in the blood and looking at antibodies, 25 to 40% of individuals at any one time. Now, it may be higher in the CSF, and then we don't know about long-term T cells. I'll share with you in a second something about the T cells. But whether the immune response to GliaCam at the antibody level is evanescent or fleeting or not, we need more information. And we're working hard on it with ongoing collaborations with the Escorial Lab and with the Karolinska. So answers will be emerging, and I imagine others in the community are also working on this. Next slide. So as I mentioned, we were able to isolate T cells. The T cells are both CD4 and CD8. Some of the T cells make both gamma interferon and granzyme. These are CD8 T cells. So the T cell story, both in the periphery and if we can get enough specimens in the cerebral spinal fluid, will be another area of important ongoing research. Next slide. So just to summarize, we've identified a molecule of interest, GliaCam, and some of the antibodies in the CSF in MS patients are directed to it. There's extensive mimicry. There's a core feature with two prolines, an arginine, and other two prolines that are exactly shared, and some other similarities. We're looking at the degree of cross-reactivity. This population is in plasmablasts, which lose their CD20, but the plasmablasts are derived from the CD20 population. So it's a work in progress, but very exciting area. And I didn't think at age 75 I'd be on another new exciting chapter. So to me, this is really going to be an interesting run for the next few years through the whole community. Thank you.
Dear Larry, thank you very much for this great presentation. We have now heard the causal relationship between EBV infection and cross-reactivity of B cells to protein structures in the brain. It would be now interesting to see whether EBV infection is only the initial trigger of MS, or whether disease progression is associated with reactivation of MS. So here, I would like to turn over to the next speaker, Dr. Heinz Wiendl, and Dr. Heinz Wiendl will now present his work on a broader EBV-specific T-cell receptor repertoire in patients with MS, which could give us an answer to the following question. To what extent is active EBV infection an ongoing process during MS progression? Dr. Wiendl, please unmute yourself and start with your presentation.
Yeah, I hope you can hear me. Very, very excited to speak about this topic, and our approach to the topic. Can the slides be shown here?
We can hear you, and we're working on the slides. Give us 2 seconds.
Okay. Let me start already by saying that we came from a completely different avenue, and those two big roads come together extremely nicely and come together, and that will be one key conclusion to a plausible and also testable readout for possibly biological correlates of multiple sclerosis activity. So our lab is extremely interested in signatures to characterize multiple sclerosis activity, to identify specific signatures both on the peripheral immunological level, but also the cerebrospinal fluid. And the difference from what Larry Steinman just brought up is that we didn't start really with B cells and antibody producing cells. Our key aim is to characterize the T cell repertoire. Next slide, please. You heard already quite a bit, next slide, after my disclosures, you heard already quite a bit about the ongoing discussions on EBV and multiple sclerosis and the extremely exciting and convincing work that EBV seems to be a trigger. But the question is really, what does it have to do in terms of maintaining ongoing disease activity? Or is it a driver? Next slide. And I'm just recapitulating the guilt by association, and a lot of that has been already done in the previous talks. I think all in all, the association is very convincing, but doesn't really explain why EBV is a essential, but in itself not sufficient condition to really drive the disease. Next slide. The papers that you have just heard about have made this causal association now extremely plausible by getting the specificity for EBV as opposed to other herpes viruses and other viruses clearer into the ground. And what Larry Steinman just brought into is a molecular characterization to convince that this infection is associated with a cross-reactivity towards structures in the central nervous system. Next slide. We are working, as I said, on the other arm of the immune system, T cells. And you certainly need T cells to recognize viral structures. I think you're all aware that the key structure to eliminate viruses is usually CD8 cells. There is a number of works that have tried to associate also disturbed T cell function and T cell specificity to Epstein-Barr, and CD4 T cells have been associated. Next slide, but also CD8 T cells, and this CD8 T cell work was in a way pretty provocative in the field because the presence of obviously CD8 EBV specific T cells in the central nervous system has brought up the hypothesis that there is ongoing viral activity bringing into play the possibility that multiple sclerosis might be a result of ongoing virus activity or cross-reactivity. And that has really provoked the field in a way that a virus kind of is not the trigger factor only, it's really a central part of MS pathology. Next slide. So the hypothesis, we actually went out, and I think the convincing part is we went out without really a strong hypothesis, because the data that I'm showing are coming from a larger, let's say, landscape project, where the part of EBV specificity was then a data set we had collected, I wouldn't say by accident, but it helped us a lot to really look specifically for antigen-specific receptor sequences. So the idea was, if there is a CD8 activity in the tissue, would that not be associated with a measurable disturbance in the T cell receptor repertoire? Next slide. So what we used here, and that's actually the complete study design, is a pretty big cohort, the so-called discovery cohort, consisting of nearly 1,500 patients and 230 controls. We had a validation cohort that was much smaller, but still validating, and we had an invaluable cohort of twins, monozygotic twins, that are discordant for multiple sclerosis. And what we did with all of them, we applied so-called deep sequencing of the T-cell receptor repertoire. And what that means is one T-cell receptor recognizes one single peptide antigen in the context of MHC1 or MHC class 2, either CD8 T cells or CD4 T cells. And you can actually sequence them all. And this is the so-called T-cell receptor repertoire. Within this T-cell receptor repertoire, you have memory responses against known antigens, and they give you specific sequences. And those sequences can be interpreted in the context of a certain so-called HLA background. And what is very well known, meanwhile, are known sequences against Epstein-Barr, SARS-CoV, CMV, another herpes virus, and also influenza. So we had the possibility with the big data set we acquired in this pretty big cohort to look specifically for the virus-specific sequences. So as I said, we went from a general unbiased approach to an antigen-specific sub-aspect. Next slide. And this is already the key result. So using two different types of immunosequences, using peripheral blood, we could clearly see that there is a difference in the repertoire between MS patients and non-MS patients, and this difference was only visible in the repertoire against Epstein-Barr virus. Next slide. And this actually was seen both in the discovery cohort, which was the bigger one, but also in the validation cohort. What does these numbers mean, and what does the quantification mean? I mean, as I said, you have many different T-cell receptors, billions, and some of them are directed and plausibly directed against Epstein-Barr. In the context of HLA-A2 and other certain HLAs, you can actually pin that down to CD8 cells specific for specific antigens of the Epstein-Barr virus. And there is different ways to quantify the repertoire, and one way is to quantify how broad the repertoire is. That means how intensively the immune system deals with the virus as opposed to a healthy immune system. And that brings it to a very strong point, because what Larry already mentioned is that 96% of patients would have also antibodies against EBV. And with classical assays, you would simply measure its positive or negative, and maybe also the quantity of the antibody. With the repertoire, you can go much deeper and much more quantifiable. So you can actually quantify the elements of how this repertoire can be described, but also how it can be changed. And what really struck us a lot is that the twin cohort, which you see on the lower right, also showed a statistical difference. So those results will never be black and white. So on a, let's say, level of many individuals, you see statistical differences, and that is done with biostatistical methods. But the result overall was extremely robust and really struck us a lot. Next slide. Furthermore, and I think that brings the story into a context of how could that help us in understanding what certain immune therapies do to multiple sclerosis and what other therapies don't do. So one therapy in multiple sclerosis is the already mentioned natalizumab, which is a paradigm for us to really investigate what is the infiltration from outside into the central nervous system. As said, very instrumental work done many years ago by Larry Steinman and Ted Yetnock, and this has now established itself to a very crucial therapy blocking leukocyte infiltration and blocking focal inflammation in the multiple sclerosis field. What you see is that you don't really change a lot in terms of the responses against SARS-CoV, CMV, and influenza. That's in the middle of the slide. But when you look upper left, you see that obviously you're increasing the number of EBV-specific T-cell receptor sequences. And that makes sense because obviously in the context of multiple sclerosis, you have an ongoing response and an ongoing attempt of the immune system to fight against EBV triggering and perpetuation. And that means when you sequester those cells out, they would increase. With other therapies that I do not show, including interferon and B-cell depletion, we didn't see much of a change. That means, on the other hand, if a therapy would change the repertoire, that would be a plausible argument to say, well, the driving force that obviously is associated to MS pathology and maybe progression can also be measurably affected. And that's the difference, the big difference of the T-cell receptor repertoire studies to antibody detection. And that makes this type of analysis an attractive way to understand MS pathology, but also the impact of certain therapies. Next slide. Last but not least, and this is a very convincing data point, we asked ourselves, we see the difference in the peripheral blood, but do we also see a difference in the type of the cells that have Epstein-Barr virus specific receptor recognition elements in the cerebrospinal fluid? And I think that is one of the most convincing data parts, because there is clear differences in the immune composition between healthy controls and multiple sclerosis patients. You just saw that plasma cells or plasmablasts can only be found in multiple sclerosis patients. And the other difference is on the T-cell side. So you see certain subtypes of T-cell receptor T-cells, T central memory as opposed to T effector memory cells. And while you wouldn't be able, based on just antibody levels in the peripheral blood, to see a difference between the MS patient and the healthy control, the MS patients really had a much more different subtype that was associated with the Epstein-Barr virus. So the phenotype of the cells we detected was indicating that there is a recent priming in the recognition of lytic antigens, which is very compatible with the hypothesis that there is ongoing viral replication. And that's a pretty strong point, because that goes far beyond the triggering hypothesis. It suggests that there is obviously ongoing activity that makes the type of the immune cells in the compartment adjacent to the central nervous system very different. Next slide. So this phenotype in the cerebrospinal fluid points toward there is expansion of central memory cells and a strong shift to recognition of lytic Epstein-Barr virus antigens. And I think that strongly suggests that there is an active immune response directed against EBV, measurable either in the peripheral immune system via peripheral blood analysis, or also in the cerebrospinal fluid that indicates there is recurrent Epstein-Barr virus activity. And next slide. So the conclusion of this story, and I think the beauty of the stories is that they come from different underlying hypotheses. So one is epidemiology. The Larry Steinman story is identifying antigens recognized basically by oligoclonal bands in multiple sclerosis. And our story comes from can we use T-cell receptor signatures, not knowing what the antigens are, to identify what is MS differing from non-MS, and then pinpointing towards certain viruses, and thereby seeing a strong signal in 3 different cohorts, including monozygotic twins, that there is a broader repertoire, means a broader immune exchange between Epstein-Barr virus antigens in MS individuals. We could identify specific sequences. We could identify an altered response to latent and lytic epitopes in 3 differing HLA-B backgrounds. And we believe that our data are compatible with the hypothesis that the priming of CSF infiltrating central memory cells is an ongoing thing in multiple sclerosis. But the key point in terms of what will we do with future studies is that we have now created a testable hypothesis to associate disease evolution also with the Epstein-Barr virus repertoire, and also being able to test how and in what extent certain disease-modifying treatments, and one example would be DHODH inhibition, modify this repertoire based also on control cohorts we have with, as I just showed you, natalizumab, leukocyte sequestration treatments, or B-cell depletion treatments. So for me, it's the first, let's say, plausible biosignature associated to multiple sclerosis that comes via a pretty hypothesis-free way and is pretty established, because we have this huge data sets on all sorts of MS patients, but many also controls. And that actually allows us to test now the impact of disease evolution, different disease phenotypes, but also different disease modifying treatments in a statistical way. And next slide is, I think, already the acknowledgement to the team. I showed you one image that is Nicolas Schwab. Nicolas Schwab was and is instrumental to this studies. He's the one that is doing adaptive immune signatures in neuroinflammatory conditions. That's a big journey where we try to generate a landscape of neuroinflammatory disorders and its therapeutic intervention. We are already quite far with this type of analysis, also doing unbiased immune phenotyping with flow cytometry. And we have used a variety of cohorts. One is together with the Immunic team, the twin cohort, and the other is with Roland-Liblot, the natalizumab cohort from Toulouse. Thanks for your attention.
Great. Thank you very much, Dr. Wiendl, for this very interesting presentation. We will now open the webcast for our first question and answer session to give the audience an opportunity to discuss with our excellent MS experts. We actually still have all three of them online. As a reminder, if you joined the webcast via the Zoom platform, there are two ways to submit your questions. You can either submit in writing via the Q&A tool of the Zoom portal, or if you would like to speak with us directly, please use the raise hand function of the Zoom portal to queue your question. And I think we have a couple of hands raised. We first go with Yasmin Rahimi of Piper Sandler. Yasmin, please unmute yourself and go ahead.
Thank you so much, Jessica, and thank you so much to all of our physicians for taking time out of your busy schedule to be with us. It would be really great if you could maybe share your comments on the data that the team presented this morning, the 96-week OLE data and the disability progression. How remarkable is this data? How does it compare to others? I know the Immunic team showed a really nice comparative slide, but I would love to hear sort of additional commentary. And then second, in regards to the EBV virus data and its link to progression of disease in MS, could you maybe highlight how could the team at Immunic, incorporate analyses so that once they have the phase 3s are completed, such data could end up in the label and they could actually drive and make the claims to be preventing these infections. And thank you again for taking my question. I'll jump back into the queue.
I think I'll take the first. So that the continuation in the open label extension data gives you confidence in the consistency and the durability of the data. It also gives you additional safety information. The information they already have is highly indicative of what we would expect to see in a phase 2 and highly indicative of what would give success in a phase 3 trial.
And I may take the second question. So one possibility, and this is something we're going to explore, is to use this first as an exploratory endpoint in a trial condition, and we're trying to convince to do that also in the context of DHODH inhibition. And if there is a meaningful change in the broadness of the T-cell receptor repertoire as compared to placebo, that would be an extremely strong hint towards two things. First of all, the mechanism of action of DHODH inhibition does something in terms of EBV triggering and EBV repertoire as a consequence of the triggering. Plus, this might be a biosignature that could be associated with the mode of action and thereby measurable. What we don't know, I have to say honestly, how much the repertoire is really associated to the term progression. So that is one question we have to answer in longitudinal cohorts. So would a change in the repertoire be more associated to disease progression as opposed to relapse biology? That can only be answered with very well curated longitudinal MS cohorts, but we have our hands on something like that. But particularly answering the question on how would that affect the Immunic product, it could be a unique feature that this DHODH inhibitor can claim that it really affects EBV replication, and that can be measured in a accessible way, means via peripheral blood.
Thank you. And then if I may just squeeze one last question. Given that you all might be involved in the ENSURE as well as the CALLIPER studies, could you comment on sort of the enthusiasm that exists for patients to be part of that study, and how you as an investigator sort of make clear the differentiation of this DHOH versus the one that is currently available on the market? And thank you again for letting me ask one additional question.
So I think I'll start again. There's great enthusiasm for CALLIPER because we don't have particularly good therapies or adequate therapies for progressive disease. And the other is that even though it's a DHODH, its mechanism of action is still different. It's a different class of drug. And so one would hope to see potentially better safety, which become a factor in thinking about relapse or remitting disease.
Yeah, very much along these lines. I think a safe and reliable player that has a good impact on relapse biology, but also progressive elements of the disease is extremely welcome in the field. And by the mode of action, it's really a next generation DHODH inhibitor. As you have seen, the selectivity and the specificity towards DHODH should result in the expectation that we have also seen in the available phase 2 programs. This is a very well working, but also very safe drug. And this is exactly what the field clearly needs, because most of the drugs have problems with tolerability and monitoring. And this is making the request of having safe players and efficacious players in the MS fields a call for action.
Thank you so much.
Yasmin. Our next guest is Matt Kaplan of Ladenburg-Thalmann. Matt, please unmute yourself and go ahead.
Thanks, Jessica, and thanks to the KOLs for their presentations today. Just a follow-up on Yasmin's question, maybe a little bit more into the weeds. I guess given what we've seen so far with Vidofludimus in terms of safety and efficacy in the emphasis phase 2 study, and the longer-term safety now in the open label extension, just for the doctors, how do you think about potentially incorporating Vidofludimus in your treatment paradigm for both relapsing, remitting patients, or even potentially, I guess we haven't seen that much data now in progressive MS, but specifically in RRMS in the future if the positive results continue?
Well, we'll need to see the full efficacy and safety results. And if they play out similar to what we're seeing in the phase 2, then it becomes a safe and effective alternative for patients to put on an oral therapy, and becomes one of the factors in choosing the therapy. There's many, many factors involved in choosing it. It's an hour-long conversation with the patients because there's many things there.
Yeah,
but that would be a valuable addition.
Exactly. The short answer from my side would also be it's an option in this broad field of treatment naive or eligible to switch relapsing populations. Many patients still prefer oral compounds. Within the oral compounds, the available ones, they all have, shorter or mid-term problems. The DHODH inhibitor, the ancestor, is actually a pretty well accepted principle because it's usually running without major problems on the long run. On the other hand, there is still concerns about some aspects of tolerability, which might not be the same with Vidofludimus. And therefore, I would agree with Fred. It's something mainly a priori, if the data come out as we expect for relapsing population. We would hope also that this effect on progression is impactful enough to maybe argue principle, because progression is also an element of relapsing disease biology, as we know nowadays.
And I guess just one quick follow-up. Given the drug safety profile, do you see a potential future of combination therapy potentially in MS and for this drug?
I might start. I mean, the real combination is to combine either available or yet to be developed products. And what I would see is a combination of this type of a mode of action with a mode of action that is rather doing remyelination or repair. So far, none of the combination therapies of two immune-acting components has really provided superior results to as opposed to monotherapies. The other aspect is, and I think that is maybe more important, is treatment sequencing. So what do you do after you have applied certain, let's say, high efficacy therapies, things like B-cell depletion? You can't go forever. So what would be an ideal sequence to follow up? And I do think that a treatment sequence is maybe I shouldn't say lower hanging fruit, but a more realistic fruit and real unmet need in the field than a combination of primarily immune acting drugs.
I would agree with that on both counts.
Oh, thank you very much.
Thank you, Matt. And thank you to our KOLs for answering the analysts' questions. I think we will go on now with our Immunic presentation. Let me reshare the slides and hand back over to Andreas to give an update on our ongoing MS trials.
Yeah, thank you for Fred, for Larry, and Heinz, because I think it's very hard to continue now with the program after this. After these presentations. I just wanted to give a small and short update about, of course, that we have our ongoing development program in multiple sclerosis that consists of a phase 3 program that we call ENSURE in relapsing MS. These are two identical pivotal trials that uses the 30 milligram dose of Vidofludimus calcium. And we really designed it to be a low-risk clinical program for getting for regulatory approval of Vidofludimus calcium in the relapsing MS population. We also, in parallel, have the phase 2 study ongoing called CALLIPER in the progressive MS population. That includes both primary and secondary progressive MS patients, which uses a dose of 45 milligrams of Vidofludimus calcium. And really here, I think the the idea is to really highlight an effect of vidofludimus calcium on the progressive part of the disease in MS in a non-relapse setting. So I think we believe that this study also has a importance even for the approval in relapsing MS, because it highlights for example, the progression independent of relapse activity, because that's a non-relapse setting in these progressive MS patients. And all of these studies are ongoing and recruiting, and we look forward to give you a little bit more updates once we have the data from these trials, or at least interim analyses. Next slide, please. Just to highlight a little bit, the phase 3 program, the ENSURE trials, these are 2 identical trials, placebo-controlled, about 1,000 patients each, with the primary endpoint in each of these studies being a relapse endpoint. And then we have the agreement with the regulatory agencies that a key secondary endpoint is done on a pooled analysis, which is to confirm disability worsening, similar to what I've showed you today, and basically the first data from our phase 2 trial. This will be confirmatory that's probably also been used then for labeling this drug as in having an effect on confirmed disability worsening. So this is ongoing. So next slide, please. So this is just a short summary of our CALLIPER trial, the phase 2 trial in progressive MS. Sorry, there's a typo here in the heading. I just see this. It's PMS, which is a 120-week follow-up study. Primary endpoint is an MRI-based endpoint, basically that evaluates brain atrophy, and we have a lot of biomarker and functional readouts here as well in this trial to really get a full picture of the effect of vidofludimus calcium in progressive MS patients. So next slide, please. This just summarizes, I think, the expected data points that we have from the MS program. We will have a CALLIPER interim analysis that we will report in the second half of next year, 2023. That includes several biomarkers, but also includes some of the functional outcomes from the CALLIPER study that we will report on half of the patients in the CALLIPER trial after 48 weeks of treatment. And then we have the full CALLIPER readout, which is expected at the end of 2024. That gives us the readout from the progressive MS study. And then we have the first ENSURE study, the ENSURE 1 study, is expected to have the readout in end of 25, and then about half a year later, the second ENSURE study will have the readout. So this is the expected sequence of our data readouts here for the MS development program. So next slide, please. This actually goes into, I think, Daniel. I will hand over to Daniel to basically pull this all together for what the drug means to our in MS and what strategy and positioning we have. Thank you.
Yeah, thank you, Andreas. And I will try to do my very best to bring things together into the point and to put it in the context of what we are doing as a company here. And there's one thing I wanted to shortly talk about. So today, I think we learned a lot regarding the origin, progression of MS, some really brand new data and hypotheses, which I think are helping a lot and will guide the way in the next couple of years to specifically improve situation of patients. Today, I would like to also use the opportunity to hint to an aspect which is important for patients, which is safety and tolerability. And there was an interesting paper published by Bob Fox and co-authors investigating to what extent patients are willing to take the risk of potential side effects of currently used drug. For example, 64% of patients with multiple sclerosis were not willing to accept the risk in this study of a treatment with less than 0.001% likelihood of experiencing PML or loss of kidney function. And also interesting from our point of view is that 47% of MS patients forgo treatment due to safety concerns. In conclusion, we should not limit our work to improving relapse reduction and preventing of disability worsening only, but also offer patients a safer and better tolerable treatment option. Considering that current treatment options come with risk, including loss of B cells, associated immunosuppression, and so forth, risk of getting PML infections or even cancer, high adverse event rates in general, monitoring requirements, and several tolerability challenges, it is not surprising that, and maybe it is surprising, that glatiramer acetate is still the second most frequently used drug in MS therapy, despite its relatively modest therapeutic benefit. And we believe that vidofludimus calcium could be a perfect solution in this context, given that vidofludimus calcium can target various aspects of this smoldering MS by combining antiviral effects we have seen in several presentations today, including the inhibition of the EB virus, a selective anti-inflammatory activity demonstrated, I think, with quite impressive data set from our phase 2 emphasis trial with very strong reductions of MRI lesions and reduction of relapses. And most importantly, vidofludimus calcium has a unique potential of a direct neuroprotective effect, targeting the key patient need for slowing down or preventing disability worsening. The new data shown today by Andreas also underline this potential with strong clinical evidence for reduced rates of confirmed disability worsening in the open label extension part of the phase 2 emphasis trial. Vidofludimus calcium is therefore addressing key unmet medical needs of MS patients in multiple patient segments. Supported by the emphasis data, the drug could clearly raise the efficacy standard for risk-intolerant patients and offer patients who are in the need for alternative treatments a promising mode of action, matching the MS pathophysiology, avoiding numerous shortcomings of currently used DMTs. Underlined by the recent data and the latest mechanistic and preclinical findings, we see a huge upside for the use of vidofludimus calcium in patients with progressive disease, where disability worsening still remains the largest unmet medical need. And finally, we're convinced that the drug could be an attractive option for patients currently untreated for various reasons. This is supported by an outstanding safety and tolerability profile generated so far. To summarize, vidofludimus calcium, with its unique threefold mode of action and its unrivaled safety and tolerability profile, stands out of the crowd of current therapies and is aiming to elevate the standard of care with a holistic solution for MS patients. Yeah, with this, I would like to open the line for another round of Q&A, also potentially directed to the company and our thoughts on going forward and so forth, and happy to take your questions here.
Yeah, thank you, Daniel. Again, as a reminder, please use the Zoom platform to queue your question, either by raising your hand or using the Q&A tool to submit your question in writing. I actually have received a couple of questions in writing. Let's start with the first one, which is, have you done any analysis from your emphasis trial about patient EBV status or flare-up reduction, possibly in a post-hoc analysis from the original trial? And as a follow-up, is there any plans for analysis for your ongoing ENSURE and CALLIPER trials?
So this is probably directed to me.
Exactly.
Yes, we have done a post-hoc analysis of the emphasis trial regarding EBV status. And I'm embarrassed to say, after you saw the presentation by Heinz Wiendl, you probably understand that this is really not meaningful, because it's really not addressing what's the status in itself, because it's such a prevalent virus, won't give you a lot of information. So what we have now done in the phase 3 trial is that we actually included, for example, the EBV shedding analysis in saliva into all patients, and we're also trying to implement some of the newer analyses that you have seen and getting samples for those analyses. However, you have to remember that this is all very recent publications, so we're kind of trying to implement this and get maybe a subset of patients that where we get these information. This is all brand new information. Everything that we heard today was published in 22, so, or even just a few weeks or months ago. So it's something that we try to address, and because I think these are wonderful ideas. These are, we believe that all of these information regarding the pathophysiology and the immunology in MS patients actually is something that could be extremely helpful to the development and to the positioning of Vidofludimus calcium. And we hope we can implement as much as possible into our ongoing trials.
Okay, let's switch on actually to a set of questions that we received from Andreas Ageridis at Wedbush. I will walk through them one by one. First one is, in the OLE phase, are baseline characteristics of the patients comparable to those in the historical controls?
That's an easy answer, yes, because like I said, we had very few discontinuations in the blinded treatment phase. And all of the patients who reached week 24 actually continued in the open label phase. So I think the open label phase, more or less, is the same patient population, minus very few that discontinued in blinded treatment phase.
Can you provide any insight into the patients who discontinued from the OLE, excluding those with MS-related events and those related to the conflict in the Ukraine?
So this is basically, you're asking me about usually other reasons that are usually labeled as other reasons to discontinue a trial. So we had patients that, of course, had MS-related events, and those that are related to the Ukraine conflict. There is most of these patients that discontinue other than these events, I think do this because it becomes that they're interested in taking another treatment, or they're moving. There's lots of reasons why patients don't do this, and there is, I think, there's very few patients who discontinued because of adverse event, but this of course happens in every clinical trial. But there's nothing specific about other reasons. Sorry that I haven't. So I didn't list all of the reasons because some of them need a long explanation, and they're single events. So there's nothing systematic in these other reasons why patients discontinued from our open label trial. And I think the rate, as you see, is probably very low. As you have about 5% of patients, a little bit more than 5% of patients per year basically leave our open label trial, which I think for these type of open label studies that's considered very low.
Thank you. And here's the third question from Andreas Ageridis. How is IMU-838's effect on viruses differentiated from teriflunomide?
Yeah, I think I was expecting that question, basically. Somehow, unfortunately, there's little known about that, and that's something we will maybe at a later time point can comment more on that. We just know that our drug has quite good in vitro activity, and we can match it nicely with the exposure we see in the patient. So that's maybe what you want to see and what you want to achieve as antiviral activity in those patients.
All right, next one is, how are you capturing IMU-838's impact on EBV and even COVID-19 in the phase 2 CALLIPER and phase 3 ENSURE trials?
And this is maybe once again for you.
Yes, I assume. So I think COVID-19 is captured as a adverse event, and we have seen already some COVID-19 infection. So that effect comes from more or less from the adverse event page, and that the data that I showed you from the emphasis trial was also basically that all of the infections that happening in these patients are captured as adverse events. So this includes, of course, other infections, other viral infections, but also COVID-19. And the effect on EBV, this is an ongoing story, yes? And I think I can just refer to what I said earlier. We're trying to implement subpopulations where we look at this. in an ongoing clinical trial, because we are learning. This is all basically work that was published in this year, and we're trying to implement this as much as we can. But I think this is an ongoing phase 3 trial, and we may not get all of the measures that we want in all of the patients, but I think we believe it's important. We highlighted this here, these data, because we think they're very important, and actually these data really support the positioning of Vidofludimus calcium. So we make a lot of efforts to implement this into in our ongoing clinical trials in MS.
Yeah, as a reminder for everybody else on the line, if you have a question, please use the raise hand function of the Zoom portal. And I have another one that came in writing. Can we connect the dots with today's presentation on disability worsening and EBV reaction secondary endpoints to the primary endpoint of time to relapse?
So I think that's probably more difficult. And the reason is, traditionally, a relapse-related or a relapse endpoint is considered a measure of anti-inflammatory activity of a drug. And in that respect, I think you have usually, and that's also, we had this in published slides earlier before, that you have a huge meta-analyses that look at, for example, the connection between MRI lesion suppression and the effect on relapse that you have in later clinical trials, and that there's a very good relationship with that. There's a correlation. There's actually even an equation where you can put in your MRI reduction and get a expected relapse reduction in the phase 3 trials. But this is all more or less related to anti-inflammatory activity. What we have discussed here today with the EBV infection addresses more kind of the silent progression or the unrecognized progression, how Fred called it, and the smoldering disease in the background that's going on. If you have relapses or not, you get an increase in disability worsening. You have this basically this slowly increase in accumulating more disability over time that may not be related to as much to anti-inflammatory activity. And there, I think the anti-EBV activity or our neuroprotective activity plays a larger role. And this is, I think, where I see more of the ways of differentiating this drug to other drugs, because we have these properties in our drug, and this translates probably in other endpoints other than relapse more so, and differentiates more so than in relapse itself. But relapse is definitely, I think, for the regulatory authorities, a necessary primary endpoint to ensure approval. And that's why our studies also had been designed and sized also to basically give you a relapse effect that leads to approval. That's, I think, number one importance, of course, of a development program in phase 3.
Thank you, Andreas. I received another couple of questions from Tom Smith at SVB Securities and would like to read them again. Can you provide any more granularity on the safety experience during the open label extension portion of the emphasis study as it relates to AEs? What proportion of the treatment discontinuations from the OLE have been attributed to AEs?
So I think I can only say we plan to have a much larger discussion of the safety and tolerability, including all of the questions, Tom, that you ask, addressed in a publication, likely Congress related. There are several opportunities now in the short term. In summary, I can say I think the data from the interim analysis for the open label certainly have confirmed, and there's the safety profile that we had shown you earlier. This drug is well tolerated, also long term. There's no new safety signal, but we will give you a more granular update on this soon. But we really wanted to focus here on the on the new understanding of MS disease and how our properties really very much align with this new understanding of MS as a disease, and also therefore wanted to give you a first hint at the a first look at the confirmed disability data that go basically along these new understanding of MS disease, what's important and what's unaddressed, because the effect on or delaying disability and especially delaying relapse independent disability is one of the major unmet needs in MS, and that's what I wanted to show here. But certainly please be assured that we will publish very granular safety and tolerability data very soon, but in a different setting, and there will be no new signal coming out of that.
Thank you. And one more from Tom Smith. Can you expand on your expectations for the CALLIPER interim analysis next year? What would you consider a home run scenario for these data? And is there an opportunity to make any modifications on the trial design based on the interim results?
Okay, this is another more difficult question. What are your expectations? We have designed this interim analysis to really understand what effect drug treatment versus placebo has on biomarkers. That includes neurofilament light chain, of course, which also has strengths and weaknesses in the progressive MS population, but also others like GFAP that was mentioned here by Fred, that is maybe more applicable to progressive MS population. And we have some functional outcomes that are part of this trial. So all of this, I think, of course, is an interim analysis. So you want to get an assurance that you're on the right track. And of course, you can always make changes after an interim analysis. I think here, in particular, I think you also want to see, because we're including primary progressive and secondary progressive patients, and so far what we have seen there going into the trial at almost the same rate, so the same proportion, that you get an initial understanding, of course, if both population, I think, react similarly. And this would be something that may not immediately be change the conduct of the trial, because the trial at that point, when we have the interim data, may be fully recruited already. But it helps us also in terms of planning for next steps, planning for other things to do, and that's, I think, also where we see the value of this interim analysis, rather than changing necessarily the design of the trial.
All right, this concludes our question and answer sessions for today. I would like to turn the webcast back over to Daniel for any closing remarks.
Yeah, thank you, Jessica. Yeah, at the end of this R&D day, I would like to express my thanks, first of all, to our speakers, Fred Lublin, Larry Steinman, and Heinz Wiendl. We are honored to have had these three leading experts here with us today. I would also like to thank all participants for attending and the lively discussion and encouraging questions and comments received. And finally, and it's very important for me as well, I would like to thank my colleagues at Immunic. It is great to feel the enthusiasm in the team and to be able to reap the fruits of long, intensive work. Here at Immunic, we can feel that we're part of something very big and novel. I wish you all a wonderful remaining day and a nice upcoming Thanksgiving week. Thank you. Bye-bye.
Thank you for joining Immunic's MS R&D Day today. The webcast has now concluded. You may now disconnect.